Hi everyone. This is the second lecture for your chapter Photosynthesis. In the last class, we discussed all the timeline of discovery of photosynthesis. How different parts of the same process were discovered by different scientists and what all methods they used for experimenting, right? We also studied a few experiments that you may or may not have done in lower standards or in labs. Okay, and today we start with the next part of photosynthesis. Continuing with some early experiments, today what is relevant to the pigments that we will be studying, there was a scientist called T.W. Engelmann. Okay, now this T.W. Engelmann used a prism. I hope you all know what a prism is. It is a triangular pyramid shaped glass object or crystal, okay, which will split the white light, monochromatic light, into 7 colors with cure. Fine. So he was just experimenting. He took this prism and he shined light on it, and it got split into 7 different colors. He illuminated a green algae called cladophora and placed a suspension of aerobic bacteria. So what he did was, in his setup, just for your information, this is your light, this is the prism, and the prism which is splitting the white light into these 7 colors, and this is his experimental setup. Okay, in this glass jar or glass box, whatever you call it, he placed cladophora. Cladophora is nothing but a filamentous algae which he placed at the bottom. Above this cladophora, he added a culture or suspension of aerobic bacteria. Aerobic bacteria, as the name suggests, they do aerobic respiration, which means respiration which requires oxygen. Clear? Now, to this setup, when he illuminated, he just waited and watched. What he found was, or what he used was, that wherever there will be higher photosynthesis, okay, in whatever light there will be higher photosynthesis seen, in those places there will be more oxygen produced. Wherever there will be more oxygen, you should be able to see more amount of bacteria, right? So this was his thought, and this is what happened. Near blue light, okay, this, can you see, this is the blue light. The cladophora which was present near blue light, it performed a lot of photosynthesis, which meant that it released a lot of oxygen. This oxygen got accumulated in this area, and that oxygen was used by the bacteria. Okay, so bacteria started moving more and more in this part of the experimental setup, okay, because there was more oxygen on this side. Same thing was shown or was seen in the red spectrum. Okay, wherever there was a red color, there was again a lot of photosynthesis, which you can come to know by this bacteria. Okay, you can see a lot of bacteria growing in the red fraction also. Very less amount of bacteria were present in the other colored light. Clear? So from this, he came to know, or he concluded, that blue and red light help in photosynthesis more. Okay, so photosynthesis, they prefer blue and red colored light, and there the rate of photosynthesis will be higher. Today we will study what are the pigments that are involved and why this is so. Okay, why the red and the blue are absorbed more. Let us study that. For that, we will study the pigments that are involved in photosynthesis. Okay, pigments are nothing but they are organic molecules. Okay, they are substances that have an ability to absorb light at specific wavelengths. So what they do, pigments are nothing but they are, let us just consider them as paints. Okay, what do paints do? They absorb light of specific wavelength. Same thing is seen by pigments. We can separate the leaf pigments of any green plant through paper chromatography. Let us understand this paper chromatography and what it is, how it can be done. For the same, you can take any plant you want. Okay, you can take, let us say, spinach. We'll just take spinach because it is simplest. So you take spinach, you put it in such mortar and pestle, Okay, and you can add some acetone or alcohol or something, some inorganic this thing solvent, sorry, organic solvent. So you put such organic solvent, polar organic solvent, and you start mixing with help of the mortar. And as you mix, there will be a sort of chutney which will form. Right? Am I right? So you make this palak chutney in acetone or in alcohol. Fine. After that has been done, use a filter. It could be a cloth and remove all the particles that did not get mashed properly. Okay. After this, you will be left with a very light green color, very light in the sense very thin, okay, green colored solution, as you are already able to see here. You take this green solution, and what you have to do is you can take a such sort of paper. You take a paper. On this paper, first look here, okay, on this paper, you put a single drop of this solution, okay, single green drop of this solution. Now put this paper in more alcohol or acetone, whatever you have used. If you do not have any of those, you can even put it in a little bit of water and a little bit of sanitizer. Okay, it will do the same function. So you put it in such sort of a test tube, okay, and in that test tube you fill the bottom with the solvent. This solvent is nothing but acetone or alcohol or acetone and alcohol and water. You place this paper such that this drop should not touch this solution, okay, it should lay a little bit above. You hang this chromatography paper. It is just a simple paper, but it is specialized for chromatography. So you hang this paper through this cork, okay, of the your beaker or of your test tube, and now you wait. What will happen is the solvent, it will start moving up by capillary action. As you may have seen, have you ever seen what happens if a drop of oil falls on paper, or if a drop of water falls on paper? Okay, specifically try and check this on your newspaper. On your newspaper, when you apply a little bit of water on one end, that water will get absorbed and will spread, okay, even upwards. So this happens because of capillary action, which also I think you have studied in fluid dynamics or fluid mechanics. So now what will happen is, with this capillary action, the solvent will move up. When it reaches this drop, it will dissolve its component. Okay, it will dissolve whatever pigments are present in this spinach juice, and it will take it with itself. As it is moving upwards, it will take that components of this spinach solution, and it will take it along with it and rise. As it rises according to the molecular weight, okay, so if the weight is smaller, if the molecule is smaller, it will be able to run farther, okay, it will reach till the top. If the weight is more, then it will move slowly and it will reach only up to a certain level, okay, a very small distance it will travel. So after doing this whole experiment, you will see that there are different colored bands which are seen. Okay, you will find different colored bands which are seen. These colors are nothing but they are representation of the pigments that are involved in the photosynthesis. Okay, the pigments that you may not be able to see directly from this spinach juice, but when you perform this chromatography, you will be able to easily say. Okay, so the bottom most will be chlorophyll B. Chlorophyll B is a little, what you can say, yellowish, greenish in color. Fine. So and it is big in size, which is why it will be the bottom most. Followed by very dark green chlorophyll A. Then you have xanthophylls, which are orange or yellow colored compounds. And finally carotene, okay, which are nothing but orange colored compounds. Always remember carotene, carrot is orange in color. So these are the pigments which are involved in photosynthesis and how you separate them. Fine. The same thing here. This is a chromatography paper that you can see after it has been done. The experiment has been done. You can see this is chlorophyll B, which is a little yellowish green, followed by chlorophyll A, which is deep green, then xanthophyll, and the smallest compound is carotene, which will be highest. Okay, it will be yellowish orange, and you will find it above at the top. So finally, you have chlorophyll A, B, carotenoids, and xanthophylls. Fine. Now let us talk about the structure of these pigments. Okay. This is the chlorophyll A. As you can see, the porphyrin ring, the magnesium atom in between. Then you have this 20 carbon long chain. Okay. This chain is present here. And the important part is this methyl group here. Okay, there is this methyl group present here. And this is the structure of chlorophyll A. Chlorophyll B is same as chlorophyll A, but it differs at this position. Instead of methyl, it will have aldehyde group, CHO. Okay, like this, there are four main types of chlorophyll, A, B, C, D, but these two are important, so only these two are shown. Okay, the structure again is not important for exam point of view, but it is important to understand how everything is functioning. Then you have beta carotene, which is nothing but the carotene that you saw here. You normally call it beta carotene. Beta carotene is this type of structure. And as I had told you in biomolecules, if you remember, when we were talking about secondary metabolites, that one beta carotene, when it breaks from exactly middle, it gives you two vitamin A molecules. Okay, so from beta carotene, that is why people tell you to eat a lot of green leafy vegetables because they have a lot of beta carotene, and also orange colored vegetables and fruits like mango, papaya, carrots. They all have high amounts of beta carotene. Okay, and this beta carotene in your body, it will be broken down to make vitamin A. So this is carotene, and then zeaxanthin is nothing but it is xanthophyll. Xanthophyll are also a type of carotenoids. Okay, these together are called carotenoids. They are just a type of carotenoids which give you yellow color. Right? So orange color, yellow color, light green, and dark green. This is the list of all the chlorophylls or the photosynthetic pigments that are involved in photosynthesis. You have chlorophyll A, which is found in all higher plants and cyanobacteria. Okay, chlorophyll A is ubiquitous, that is, you find it everywhere. In every organism which is doing photosynthesis, chlorophyll A is present. Then comes chlorophyll B, which is present only in higher plants and green algae. It is not present in bacteria. Then chlorophyll C, which is present in diatoms. These are the diatoms. Okay, they are unicellular autotrophic organism. Okay, they are somewhat like algae only. So these are diatoms, but they have this skeleton, okay, of silica. Their skeleton is made up of silicone. Clear? So these are the diatoms. Then dinoflagellate again is a protista. Fine, and it is called dinoflagellate because it has flagella. So this dinoflagellates also have chlorophyll type C. And then brown algae. Okay, brown algae also have chlorophyll type C. Chlorophyll D is present in red algae, rhodophyceae, right? So here you have red algae which will have chlorophyll D. And finally, bacteria chlorophyll is present in purple and green bacteria, which we studied in Cornelius van Niel's experiments. Okay, so what these bacteria have is called bacteriochlorophyll. What you see here are the green algae on rocks, and this is a Google Maps picture. Okay, but in this picture, this lighter bluish green color, can you see turquoise color? That color, you will be surprised to know, is actually presence of cyanobacteria. Okay, in this blue water, the presence of cyanobacteria makes it look lighter blue or turquoise in color. These are also called algal blooms, if you have ever heard of it. Right? So these are the different chlorophylls and where they are found. Now let us talk about the spectrum of chlorophyll and carotenoids. Spectrum is what wavelength of light do they absorb, and what kind of properties they have. Normally, when you talk about optics, okay, you must have known this phenomena that whatever colored object you see, for example, here you are looking at a green leaf, it is appearing green because it is absorbing all other light except green, right? So, for example, here you have three incident lights, okay? You have green, red, and blue. So this blue colored light is being absorbed, red colored light is being absorbed, but the green colored light is being reflected. What you are seeing is the reflected light, and that is why this leaf appears green. Clear? So, right, these are the three graphs which are very, very important. Just pay attention what they are trying to say. On the y-axis in the graph number A, you have absorbance of light by chloroplast pigments. Okay, at what wavelength? Okay, on the x-axis you have wavelength of light. At what wavelength these pigments are absorbing? Wherever you see the peak, that means that is the wavelength where they are absorbing the most, and that is their favorite wavelength. Where they are absorbing less, you will see it as a flat curve here. Okay, so let us start. You can see chlorophyll. This blue colored one is chlorophyll A. It is absorbing more in blue region, which is nothing but around 400 nanometer, 400 to 500. Then it is absorbing almost nothing, little bit, and then after 650 or 620 mark is crossed, that is 620 nanometers, the light starts to become orange to red. So in red light, chlorophyll A is absorbing more. Clear? So these are the peaks, one in blue and one in red. Same thing you can see in case of chlorophyll B, which is this green colored diagram. Okay, chlorophyll B is also absorbing more in blue region than nothing, and then it will absorb more in the red region. Carotenoids are absorbing more from blue to orange, and then they are not absorbing anything. While so these are the three chlorophyll A, B, and carotenoids, the three pigments, and which colored light or which wavelength of light they are absorbing. I hope you know 400 to 700 is a visible wavelength range. Okay, 400 means it is closer to blue, and 700 means it is closer to red. From 600 to 700, you mostly see are orange to red colored wavelengths. Okay, and 400 to 500 are mostly blue to green colored wavelengths. Clear? Now, along with this graph, just to see the rate of photosynthesis, which is measured by oxygen release. Okay, just like T.W. Engelmann, who measured the release of oxygen, okay, which was nothing but indirect measure of how much photosynthesis is occurring. So here also we can do the same. We can measure the oxygen which is released, and you will find that the oxygen released around 400 to 500 nanometer is high. Okay, which means there is a lot of photosynthesis happening in this region. Then it goes down almost at around green, it will be the least, because plants do not really photosynthesize under green light. Okay, they reflect all the green light. So how will photosynthesis take place in green light? And then they start to increase wherever there is increased absorption of red light. Okay, 600 to 700 nanometer or red light. When you superimpose the two, you superimpose the absorption curve of chlorophyll A and this oxygen evolved or the rate of photosynthesis, um, you get this kind of a graph. Fine. So you can see that wherever the chlorophyll A is absorbing more, whatever wavelength of light chlorophyll A is absorbing more, you are seeing more photosynthesis occurring. It is true in case of blue light also, as well as in case of red light. Okay, so this is showing a correlation. It shows that just because they're absorbing in blue light, red light, oh, nothing. No, it is not like that. Okay, you have to always compare And when you compare, you'll find out that yes, these two processes are actually attached. Okay, they are interrelated. That absorption, wherever there is more light absorption, you find more rate of photosynthesis, which means these are the light which are preferred by the plants or by chlorophyll for photosynthesis. Wavelengths at which there is maximum absorption by chlorophyll A show higher rate of photosynthesis. We can conclude that chlorophyll A is the chief pigment which is associated with photosynthesis. Okay, chlorophyll A is the main boss. Whatever it does, everything else will follow. Chlorophyll A is the major pigment responsible for trapping light in blue and red light. Okay, so in blue and red light, most of the energy in form of light will be absorbed by chlorophyll A. Chlorophyll B, xanthophyll, and carotenoids, they absorb light and transfer energy to chlorophyll A. So from this diagram, you can see that they have different absorption spectra. Okay, so wherever this chlorophyll A is not able to absorb well, the other pigments will absorb and transfer that energy to chlorophyll A. Clear? Hence, they are called accessory pigments. Okay, they are helping in the process. They enable use of wider range of incoming wavelengths of light. So just think about it. If there was only chlorophyll A, it would not be able to absorb all the energy that is falling on the leaf by sunlight. Okay, it can only absorb blue and red light. Here, because of chlorophyll B, xanthophyll, and carotenoids, it is able to get energy even from other wavelengths, which it itself is not able to absorb. Carotenoids, again, one more thing that they do is they protect the chlorophyll from photooxidation. Okay, oxidative damage is repaired or is avoided by carotenoids. This is also the reason why, if you know that anti-aging creams or anti-aging medications, they will always have vitamin A or retinol. Retinol is its scientific term. So such kind of medicine or creams, they have retinol. Even when if you go ahead and study somewhere, you will find that in wound healing, that is when you have some damage to your skin and when the skin has to heal, at that place also vitamin A is much required. Clear? So this vitamin A or carotenoid, they are also acting as uh, what you call avoid photooxidation. Okay, they act as scavenger. They help against the free radicals of oxygen. Now just some basic anatomy of the leaf, so that before we begin with the photosynthesis, you know a little bit about what we are talking or which part we are talking about. Fine. This is a normal dicot leaf. How do I know it is a dicot leaf? Because you can see two types of mesophyll cells. Okay. In case of monocot leaves, it will be equal. Okay, it will be very even in the center. But here, because you are able to see two types of mesophyll cells, you know that it is a dicot leaf. You start with the outermost layer, which is called a cuticle. It is non-cellular, which means it is not made up of cells. It is just made up of waxy material. Okay, and that waxy material is doing what? It is just avoiding water loss. It will not allow water to be lost from the top section. Followed by the cuticle is the one single layered epidermis. Wherever you hear the word dermis, it means skin. Epi means upon. Okay, so this is the uppermost layer and it is nothing but the skin or protective layer of leaf. Below epidermis, one more thing. Epidermis does not have chloroplast. Okay, there will be no photosynthesis happening in the epidermis. We shall see this when we discuss transport in plants. Okay, that there are only few cells of epidermis, especially the guard cells, which have the chloroplast and can do photosynthesis. Otherwise, the cells of epidermis do not perform photosynthesis. Then, underneath the epidermis is this column-like cells. These column-like cells are called palisade mesophyll. In general, this mesophyll tissue or mesophyll cells are the cells which have a lot of chloroplast and they perform the task of photosynthesis. Okay. So you have this topmost, on the topmost layer, you have palisade mesophyll cells, which are column-like, followed by spongy mesophyll cells. They are called spongy because there is a lot of space between them. Okay, there is a lot of holes and cavities in between these cells. So these are the spherical shaped spongy mesophyll cells. Apart from spongy mesophyll cells, you will even find some vascular bundles. Okay. Then you have this lower epidermis, which is on the bottom part of the leaf, okay, underside of the leaf. You can see some stomata here, which means there will be opening for, which will allow for carbon dioxide to come in and the water from transpiration to go out. Okay. So this here is the basic structure of a leaf. Whatever photosynthesis we will study will be mostly focusing on this mesophyll cells, okay, and how they do the photosynthesis. The chloroplast, they align themselves along the wall. Okay, in these cells also, if you focus on them, then chloroplast, they align themselves according to the light. Okay, how much light is present according to that, such that they get optimum quantity of incident light. Whatever light is falling on them, they should be able to receive optimum, which means a fair amount of light. For example, here you can see when there is light, okay, so there is enough energy, enough light energy falling on the leaf. In such cases, the chloroplast will be on the side. Okay, this is the top view, how you can see that the chloroplasts are present on the side, and this is the side view, okay, where you can see that the chloroplasts are present near the side walls, okay, the lateral walls. Now, in case of dark meaning there is not enough light falling on the leaf. So you need to make use of all energy or all light that might be falling on the leaf. In such cases, these chloroplasts, they move upside, okay. So you can see a lot of chlorophyll gathered here, okay. And this is in this oblique view or side view, you can see chloroplast will be present here, okay, to get more and more amount of light. So these chloroplasts also, they align themselves in accordance to the light. When we discuss the structure of chloroplast, which we have already done in case of cell, but just a small recap, this is how the cells look like, okay, and in these cells you can see the green spherical portions are nothing but chloroplast. On looking at the ultrastructure of chloroplast, they are made up of two systems, okay, they are made up of two parts. There is a membranous system, which means there is some membrane which will be enclosing some parts, okay, and then there is stroma. Stroma is the liquid portion of the chloroplast. The membranous system consists of granule lamellae and stromal lamellae. Again, let us just focus on this diagram and we will go through all of these systems one by one. The chloroplast is a double membraned structure, which means it has two layers, two membranes. Okay, it has this outer membrane followed by inner membrane. Then inside you can see whatever this is, it is called stroma. Stroma is nothing but it is just a matrix, okay, in which a lot of things will be dissolved. In the stroma, these yellow colored structures that you see are 70S ribosomes. Then the red colored structures that you see are the chloroplast DNA, okay, the chloroplast genome. Then you have these blue colored structures which are starch grains, okay, they are nothing but they are accumulation of starch. Now let us come to this membranous structure inside the chloroplast. This single disc-like structure is called a thylakoid. This thylakoid is covered by a single membrane. Here it is a granule thylakoid or granule lamellae. Okay, in these cases because it is forming a stack of thylakoids which is called granum. Clear? A stack like coins of thylakoids is called granum. These are the granule thylakoids, this single one. And then there are stromal thylakoids or stromal lamellae. These thylakoids connect the two granum. Okay, they are nothing but they are interconnection. They are like bridges between the two granum. Here, so this is the basic structure of a chloroplast. If somebody asks you, whatever the pigments we have studied, the chlorophyll, A, B, then xanthophylls, carotenoids, where exactly are they present? Then the answer is, if you take this chloroplast interior, okay, you can see the thylakoids. If you take the thylakoid membrane, okay, the lipid bilayer, if you take that membrane and you zoom it and you look at it, then you can see these are the phospholipids, and in between this membrane, in between these phospholipids are these pigment molecules. Okay, these are nothing but chlorophyll molecules. So chlorophylls are present on your thylakoid membrane. Okay, they are not freely moving around in inside thylakoid or in stroma or in chloroplast. No, they are fixed onto this thylakoid membrane. It is very, very important for you to understand that chlorophyll molecules are fixed inside the thylakoid membrane. So these are the chlorophyll molecules, and here you can see this is the porphyrin ring, and this is the side chain, the hydrocarbon chain. Here, if it is methyl group, then it will be chlorophyll A, and if it is CHO group, which is aldehyde, then it will be chlorophyll B. Clear? Right. So where does the photosynthesis take place? We're talking about all the structures and we're talking about different lights and pigments. So where exactly is this photosynthesis taking place? Is it taking place in thylakoid? Is it taking place in stroma? So the answer is it takes place in both. Okay, it takes place partly in the membranous system, okay, which includes the grana, the thylakoid, and the stromal lamella, and part of it takes place in the stroma. In the membranous system, it is responsible for trapping light because we saw in the membrane itself, thylakoid membrane, you have chlorophyll. So it is responsible for trapping light and for synthesis of ATP and NADPH. You can see here as well, these are the grana, and from this grana you can see ATP and NADPH being made. Okay, so by using light. Okay, so it is making ATP and NADPH. It is directly dependent on light. If there is no light, this reaction will not happen. These are called light or photochemical reactions. Okay, they are considered under light or photochemical reactions. These photochemical reactions are the ones which help in production of ATP and NADPH. Then in stroma, there are enzymatic reactions which take place to synthesize sugars. So this is what we talked about in grana. They will convert the light energy in form of ATP and NADPH. The stroma, what it does is Calvin cycle. Okay, this Calvin cycle, what is this Calvin cycle? It will take carbon dioxide, it will use ATP, it will use the electrons and hydrogen from NADPH, and it will produce sugar. Okay, it will synthesize sugar and give out ADP and NADP plus. These NADP plus and ADP are again utilized by your grana and converted into ATP and NADPH. So this cycle continues. Clear? So enzymatic reaction which synthesize this sugar take place in stroma. Okay? These are not directly dependent on light. As you can see, there is no light required for these reactions. Just ATP and NADPH is required. So these are called dark reactions or Calvin cycle. So this is how it looks like. Okay, you can see these are the thylakoid membranes and these are the photons of light. As you must already know that light can exist in two forms. It can exist in wave form or it can exist in photon form, which is particle. Okay, it has dual nature, particle nature as well as wave nature. So here what you are seeing is a photon falling on the thylakoid membrane, and on the thylakoid membrane you have the chlorophyll and other pigments which will use this light and produce ATP as well as NADPH. What we will start with next time is light reactions, which are also known as Hill reaction or photochemical phase. Because there is involvement of light, you call it photo, and there is synthesis of chemical. Okay, from light it is getting converted into chemical energy, so you call it photochemical phase. In earlier times this was also known as light reaction. Okay, you can still call it light reaction, and it is also called Hill reaction. Just very briefly, if we discuss these light dependent reactions, what all happens in this process. There is absorption of light. Okay, so light absorption takes place. There is splitting of water. So as you can see here, H2O. This H2O will be split into protons, which are H plus, electrons E minus, and oxygen, which is O2. So in this light reaction only, you are getting oxygen from photosynthesis. Okay, this is the first reaction where you will see you are actually getting the product. Then there is oxygen release, which we discussed, and formation of high energy intermediates by different mechanism, as we will study, like cyclic and non-cyclic photophosphorylation. ADP, which is adenosine diphosphate, is converted into ATP, adenosine triphosphate. Okay, this adenosine triphosphate will later on be utilized to make sugar. Same thing will happen with NADP plus, okay, nicotinamide adenine dinucleotide phosphate. This is a what you can say, reducing equivalent, or you even call it coenzyme if you want to, right? So NADP plus will receive electrons, it will receive proton and get converted into NADPH. This NADPH will donate its electrons and protons during the dark reaction to reduce carbon dioxide. Okay, and this is how the photosynthesis process takes place. Next time we will start with the light reaction and the dark reaction. Let me check if we have time. Actually, just let's do it next time. Okay, fine. See you later.